Alrighty, welcome to chapter 13, where we're going to talk about the blood. Why are the blood and the lymphatic system paired together for this chapter, or for this module, excuse me? Well, if you remember from your first semester of anatomy and phys, you ended with the chapter of endocrinology. And endocrinology is the study of all those hormones and chemical signalers and how they travel from one organ to another. And usually they have to travel pretty long distances. And in order for them to travel really long distances, they have to have some mechanism to get there. And so usually that's the blood. And then the lymphatic system is tied directly to the blood because a lot of the properties in the lymphatic system that actually help protect us as part of like the immune system will travel alongside other cells within the blood. So that's why they're paired, and that's usually why we transition from endocrine system straight into blood and lymphatic system. So we'll start here with the blood. First, blood is actually a specialized type of connective tissue, which seems a little counterintuitive, like, okay, it's just a liquid, but it's actually considered a connective tissue, and it comes in two parts. The majority of it is actually plasma, which is 55%, and plasma basically contains water and some proteins and some really, really small sugars. Then the other 45% are the formed elements and the formed blood cells. And the formed blood cells include three different types of cells. So erythrocytes, which are the same things as red blood cells, leukocytes, which is the same thing as white blood cells, and then thrombocytes, which are the same thing as platelets. So we'll use these terms interchangeably throughout this chapter. It's really important to be familiar with all six of these terms and know which ones match with which. So if we were to take a vial of blood that was just recently donated or just recently taken from a patient, and we were to spin it down or just let it sit for a couple hours, it would actually separate into the formed elements being the bottom 45% and the plasma being the top 55%. This is just simply because the formed elements weigh heavier than water and the plasma proteins, so that's why they're going to sit towards the bottom. And this picture actually doesn't show you everything. There's actually a layer right in between the two that we call the buffy coat. And the buffy coat is going to contain some really specialized white blood cells, while the rest of the buffy coat is going to contain the white blood cells, which is part of the 45% of the formed elements. If we were to separate out what's actually within the 45%, we have the erythrocytes, like I said, those are the red blood cells, the thrombocytes, which are the platelets, and then we have the leukocytes, which are the white blood cells, and those come in 5 different flavors. One type is neutrophil, the next is basophil, the next is eosinophil, lymphocyte, and monocyte. And I think there's an acronym to remember all of these. So never let monkeys eat bananas, I believe. Never let monkeys, so neutrophils, lymphocytes, monocytes, eosinophils, basophils. Never let monkeys eat bananas, which is kind of funny. So each of these cells are going to have individual functions. They're easily identifiable under a scope, where you can identify by the nucleus or the staining of the cell or the size of the cell to identify what type of leukocyte it is. And they all have different functions, and they all come in different quantities within the blood. So neutrophils are going to be in one of the greatest quantities, followed by monocytes and then lymphocytes, while basophils and eosinophils are going to be in smaller quantities. And I'm sorry, one thing. We'll talk about the functions of each of these cells whenever we get to the lymphatic chapter, because these are the white blood cells, which are going to function as part of the immune system. So we'll talk a little bit more in detail about what they each individually do in the next chapter. So functions of the blood. What's the whole point? So the functions of the blood are to transport oxygen, O2, which is a gas, CO2, which is also a gas, nutrients, waste, and hormones. So O2, we want O2 to get to the tissues. When the tissues use O2 and glucose, one of their byproducts of that cellular metabolism is going to be CO2, and we have to have a good, efficient way of getting that out of the tissues so that it doesn't make the environment toxic for the cells. Nutrients, so we can get them to the tissues. Waste, so we can get them away from the tissues. And then hormones, so we can tell the tissues what to do. Regulation of, the blood also is one of the functions of blood is to also regulate certain body mechanisms. So one is body pH, and we'll talk, pH, as you remember, has a range of 0 to 14, with 0 being mostly acidic, and 14 being mostly basic, and 7 being around neutral. But the actual neutral of a human is around 7.35 to 7.45. So it's just a little bit above 7. It also regulates body temperature. How does it regulate body temperature? Well, as heat is trying to move within a system, if it gets trapped somewhere, then it will continuously, you'll get more and more heat to that area and you can overheat. But blood allows heat to actually travel within the vascular highway and dissipate to other tissues and hopefully to the outside environment. How the body pH operates, or how the blood contributes to maintaining the body pH at a normal level. We'll talk a little bit more in detail whenever we get to the kidneys. The kidneys are primarily function with this, and the respiratory system is also tasked with making sure that the right protons are exiting or entering at any given time, so we can make this a balanced system. So that's why I'm not going into detail about it here. Then the function of the blood is also clotting mechanism. So all those platelets, those thrombocytes, we've been taught for many years that their primary function is clotting. And that's actually only 10% of what platelets are able to do and what they actually do. So, but here we'll talk about what that 10% looks like. Protection against foreign microbes and toxins. So the blood is going to help white blood cells travel within it in order to actually target these foreign microbes and toxins. And again, we'll mention that in the lymphatic chapter. And then finally, osmosis, or the movement of water. So the diffusion of water from one area to another. We constantly need to be moving water at any given time. Our body is made up of 70% of it. So it makes sense that we need to have some sort of mechanism in order to keep it moving so that the nutrients within water can also travel. Water is super, well, I don't know if I should say super, but the human body was designed pretty smart, with 70% of it being water. Water is polar. Water is able to easily dissolve solutes, and then it's able to easily carry them from one place to another. Water isn't heavily charged, so it doesn't really mess with other tissues. And it's really small, so it could easily diffuse through the types of tissues. So when we talk about osmosis, we're just talking about the movement of water, but understand that water is also going to contain other nutrients within it. And that's why we really care about the osmosis, is because what else is actually traveling in the water as well. All right, so how do we classify blood, right? Are you A positive, B negative, and what does that even mean? Which we'll get to also. So a classification of blood cells. So we have red blood cells, which are the majority of the formed elements, or the majority of that 45%. Then we have the leukocytes, which are the white blood cells, and they come in 5 flavors. Like I mentioned, neutrophils, eosinophils, basophils, monocytes, lymphocytes, or never let monkeys eat bananas. And we can actually subdivide those 5 types of cells into granular cells, or agranular. Remember the prefix A. Remember the prefix A in front of anything means without. So the granular ones have granules. The agranular ones do not have granules. And we have some pictures of that here later. And then the thrombocytes, which are the platelets. Those are also in high abundance. And like I said, they're mainly, or we'll focus on their clotting mechanisms here in this course. All right, so some fancy pictures. So up here we have some red blood cells, or erythrocytes. This is a top-down facing view. This is a side view of the red blood cell. And you'll notice the shape is spherical and kind of looks like it has like this disc in the middle. So it almost looks like a donut, but the hole wasn't completely cut out of it. And you can also notice that they're nice in these like flat, nice looking discs. This is super important because by the time we get to the really small blood vessels, which are the capillaries, we're actually moving one blood cell at a time through those vessels. That's how tiny they are. And we need to be able to make sure that these cells are flexible, bendy, and easily stackable at any given time, so that we can put the most amount of blood in one space, and we can make it as compact as possible. So that's part of the reason why the shape of the red blood cell is the shape that it is. Then platelets are really small in comparison to the red blood cell. Under microscope, you might just see them as like tiny red dots. You might just see them as tiny like ghosted out features. They're kind of difficult to tell under a slide. Like I mentioned, we're going to talk about their clotting features here, but they have other functions as well. You might have heard of PRP injections, or platelet-rich plasma. We're not going to quiz you on that in this course, but I think it's important to understand. We have platelet-rich plasma that's either leukocyte-rich or leukocyte-poor. And what do I mean by that? Well, we either have a bunch of white blood cells or we don't have a bunch of white blood cells. And these have been used for a variety of reasons. They've been used in the plastics world, so like plastic surgeons, for reconstructive properties. They've also been used in the dental world for helping tissues, I want to say like rehabilitate better in the mouth. And we've also used them in orthopedics for injections directly either into the tendon or the muscle or the joint space. The intent is to rejuvenate and build in more tissue or build in good tissue. And this is coming directly from the patient, so they shouldn't have any sort of allergic or immune response to it because it's their own cells. However, science actually says that, or researchers have said that it only really works like 50% of the time to relieve pain for people. I'm not really sure about the stats for plastics or dental, but in orthopedics, sometimes it works, sometimes it doesn't work, and they're relatively pretty expensive injections because in order to get just a solid sample of platelets, you have to spin it a couple times and you have to get a lot of blood in order to just get a bunch of platelets. So, because remember, they're one of the smallest proportions of the formed elements, not like red blood cells, which are the majority. Anyway, moving on to white blood cells. Like I said, we have 5 different flavors. Here's a cartoonistic depiction of what's going on. And the purpose of the cartoonistic depiction is to identify the color and the nucleus differences and the granulocyte differences. So I'm going to start with the monocyte because those are the easiest ones to identify, in my opinion. These are called monocytes because they are big eaters. Sometimes they're also called macrophages. And macrophages are monocytes. So one monocyte cell. And it's named that because it has a really big nucleus, one single big bilobed nucleus, and then quite a lot of cytoplasm surrounding that nucleus. Versus the lymphocyte looks similar, except the nucleus isn't as big, and it's definitely not bilobed, and there's just not as much cytoplasm in between the plasma membrane and the nuclear membrane. The lymphocyte, the reason why we have a really, really big nucleus is because this lymphocyte is eventually going to turn into T and or B cells. Okay, and we'll talk about that in the next chapter. Then we'll have let me start with neutrophils, actually. So neutrophils are named neutrophils because they have many nuclei. They might be bilobed, they might be sandwiched all together, but you definitely cannot identify, like, there's a big difference between one single nuclear membrane versus like a cluster mess of a membrane. And this is way more likely to be a neutrophil than this. So you can easily identify those by the multiple nuclei. And usually there is a little bit of staining because they are one of the granulocytes, but not a lot. Then we have, let me actually make sure that I said that correctly. Are they one of the nuclei? Right, they are one of the granulocytes. Okay. Then we have the eosinophils and the basophils, and these are the smallest in quantity and sometimes the most difficult to find under the slide. The eosinophils are going to have one nucleus, but it'll be bilobed, might be sandwiched next to each other, but these cells are definitely going to be much smaller than the neutrophils, so that's how you can identify them. Also, the eosinophils are named eosinophils because they are stained with eosin, which is a type of reddish pink dye. So usually they look red or pink under the microscope. Versus basophils, they're named basophils because they're stained with, I forget the name of it, but it's like a blue type of dye, a blue dye. And this actually is going to look, did I even spell dye correctly? No, dye, there we go. Basophils are going to actually look blue under the microscope. And basophils won't have quite like a bilobe nucleus. They're going to have kind of like a cluster mess of what a nucleus might look like. So it might look bilobed, it might look like one stuck with another one. But really the best way to identify the difference between the basophils and the eosinophils is between the colors. And like I said, they all have different functions, which we'll get to. All right, so what's happening in the plasma? Like I said, plasma is majority of water. The rest is some really small proteins and some ions that are floating around next to the proteins. These proteins are important for a couple of different functions throughout the body, and they all have different purposes within different systems of the body, so it makes sense that they're going to travel in the blood system so that they can get to all the other systems. Some examples of plasma proteins include albumin, globulin, and fibrinogen. We'll talk about fibrinogen in the rest of this chapter. Plasma solutes are the next smallest percentage, and these are the smaller percentage also because they're really, really small. These include ions, so like sodium, calcium, chloride, etc., etc. Nutrients, so like electrolytes, vitamins. That's it. I can't think of anything else. Waste products, so like CO2 or carbon byproducts or other carbon byproducts of other cellular metabolism. Gases like O2, CO2, enzymes, which are in abundance, and you have many, many different versions of them, but those are a type of protein, because remember, enzymes are a type of protein. And then hormones, which are a little bit bigger, but they are going to be those chemical signalers. All right, so how do we even make blood cells? The process is called hematopoiesis. So anything with heme at the beginning of it is going to refer to blood, and poiesis means the creation of. So where do we do this? We do this in the red bone marrow in a majority of the bones. And we create not only red blood cells, but we also create white blood cells from the red bone marrow. So this is why we're talking about lymphocytes here and monocytes. And excuse me. Let me take that back for a second. So we produce the red blood cells in the red bone marrow, and we also begin the production of white blood cells in the red bone marrow, but some of them get specialized or finally told that they're going to become the type of cell that they're going to become outside of the red bone marrow. And some examples of those are the lymphocytes and the monocytes, which will be told by the lymphatic system what to become. How do we do this? We have to start with a stem cell. And a stem cell is going to be an undifferentiated cell that hasn't yet been told what it's going to do. So these stem cells, for example, if we're looking at red bone marrow, those cells are going to be pre-programmed to be like, okay, I know at some point in my life, I'm going to become either a blood cell or a red blood cell or a platelet or a white blood cell or a bone cell or a cartilage cell. But I'm not actually quite sure which one I'm going to be yet. I'm going to wait until I get a signal that tells me which one I'm going to become. So they're not true stem cells in the way that you think of like fetal stem cell research. Those are true stem cells that have absolutely no idea what they're going to become. They could be a heart cell, they could become an eye cell, they have no idea. These are stem cells that have some sort of linearity, they have some sort of pathway that they're going to go down. They just don't know their end result quite yet. So that's why we call them mesenchymal cells, because they have some differentiation to them, and they're going to go down this mesenchymal pathway. Okay, I already mentioned some of this with the erythrocytes with the pictures. So the red blood cells are going to be biconcave discs. So if we were looking at it from the side, it might look like this. Actually, that's not a good picture. It might look like this. So they can stack on top of each other. Or if you were looking on the top of it, you might see the kind of flattened part there in the middle. Erythrocytes also do not have a nucleus. These are the only cells in the human body that do not have a nucleus. Why? They don't really need one. Their only function is really to hold on to CO2 or hold on to O2 and deposit it, and then hold on to CO2 and let go of it. And it'll do this with some proteins, which we'll talk about here in a second. The red blood cell also doesn't live for that long. It only really lives for like about 120 days. So in 120 days, I mean, what are you really going to do with that amount of time, right? Like there's no, you don't really live long enough in order to really have any sort of cell metabolism. And if you don't really have any functions that you need to carry out, then there's really no reason for you to have a nucleus. And there's no reason for you to carry around DNA so you can carry out those properties. It would just be really inefficient for the body to make red blood cells all of the time and bother putting in DNA whenever the cell is going to die off anyway. So that's why we don't have a nucleus in there. And then how does it even carry these gases? How do we even do that? It does that with something called hemoglobin. And hemoglobin is usually abbreviated HB. And the hemoglobin molecule has 4 different nods, like 4 different little protein groups in the middle of them called hemes. And those hemes, I'm going to abbreviate H here, those hemes are able to actually bind onto O2. So O2 can come and latch onto one of those hemes. And so at any given time, a hemoglobin molecule might be bound to 0, 1, 2, 3, or 4 oxygen molecules. So if it's bound to 4 oxygen molecules, we call that fully saturated. If it's bound to 0 or 1 molecules, then it's not saturated, so it would be unsaturated. Now, these heme groups are right next to proteins, and those proteins are going to be the globulins, and those can bind onto CO2. So if it's not bound to O2, it's likely going to be bound to CO2, but not, it's not going to ever be bound to 8 things at once. It'll only be bound to 4 things at once because there's not enough room for all 8 things to bind. There's only enough room for 4 things to bind. So it could be like 2 O2 molecules and 2 CO2 molecules, or 1 O2 molecule and 3 CO2 molecules, or some combination thereof. And hemoglobin can even also bind to glucose, which is one of the ways we can actually diagnose diabetic patients. So hemoglobin has a lot of different abilities, but what we primarily care about in this course is the ability to bind to O2 and CO2. All right, let's talk a little bit about what the function is of those leukocytes, or those white blood cells. Remember, I said that they come in 5 flavors, but we can separate those 5 flavors into 2 big groups, granulocytes and agranulocytes. So why do we care about them being granular? What does that even mean? Well, under the microscope, whenever you stain them, you'll see the granular ones are going to have a bunch of granules within them. And those granules are each going to have some sort of function. And usually granulocytes have some sort of like attack mechanism to them, where they can flood a pathogen or a disease cell and destroy it in some mechanism. Versus the agranulocytes aren't going to have any of that. They're just going to probably have the nucleus in the middle. So, given that will help us remember the function of the neutrophils, eosinophils, and basophils. So let's go ahead and start with the neutrophils. So the neutrophils are one of the most abundant type of cells in the blood, and they primarily function to phagocytize foreign substances. What does phagocytize mean? Phago means to eat, and cyte means cell. So we're eating cells. And neutrophils are really good at targeting any type of cell. They're not super specific, and so that's why we have a really big abundance of them in the bloodstream, so that we don't have to constantly be producing really specific cells in order to target pathogens. We're constantly exposed to pathogens in the environment, so it's important that we have some sort of immediate mechanism to get rid of pathogens as soon as possible. Eosinophils and basophils are going to be the most, these are going to be the ones I think that are the easiest to remember, that they're granulocytes because of how they function. So eosinophils are going to release their granules, and those granules are going to be antihistamines. So what does histamine mean? Histamine is a pro-inflammatory. It's a pro-inflammatory cytokine. And the cytokine is a chemical signaler, a chemical messenger that tells nearby cells, hey, we need to bring a lot of blood here. And by making it antihistamine, that means we're not having this pro anymore. We're not bringing any inflammation to the area. And what's the point of inflammation? The point of inflammation is to increase temperature. By bringing a lot of blood to the area, now you're bringing a lot of heat to the area, and pathogens usually really don't like really hot temperatures or really cold temperatures, so usually they die. Part 2 is whenever you bring a lot of blood to the area, now you're bringing in a lot of white blood cells to the area. So you're recruiting other white blood cells that might be even better at killing that pathogen than the eosinophil might be. So by not bringing all of this over to the pathogen, or the virus or the bacteria, whatever it is that you're trying to target and kill off, then you have less of a chance for that pathogen to survive. You have less of a chance of that pathogen to survive because it won't have any of those. It won't have any of those additional cells for it to feed off of. And eosinophils typically are targeting parasites. So that's why we don't have too many of them in the bloodstream at any given time, because luckily we're not usually infected with parasites. but whenever we do have eosinophils, and they will come to the site of, will come directly to the parasite. So let's say this, I have like a worm parasite here, and the eosinophil will literally dump all of its antihistamines right around it. Okay. Then we have basophils. Basophils, they stain blue. They also are going to have those granulocytes. And those granulocytes, instead of producing antihistamines, they're going to be producing heparin, histamine, and serotonin. So histamine, that's definitely pro-inflammatory. Serotonin is a type of chemical messenger that will talk to different types of cell receptors and produce different cell responses. And we don't really go into details about what heparin is in this class. Okay. Agranulocytes, so no granules. So we don't have any granules. Granules. Whew, sorry. We have two different types of cells, monocytes and lymphocytes. Monocytes, like I said, are those really big eaters. And lymphocytes are the ones that have... they're going to eventually turn into the B and the T cells. So big eaters, they also are going to be phagocytizing bacteria and cellular debris. So they're really, really big, so they have enough room to get rid of all of that cellular debris. And sometimes they're called macrophages, and those macrophages are specific to living in the tissues. Those lymphocytes, referring to the lymphatic system, site meaning cell. So these are going to eventually turn into the T and the B cells, and we'll talk into more detail about what those cells do when we get to the lymphatic system. Okay, what about those platelets or those thrombocytes? They are also disc-shaped, but they are just fragments of cells with a tiny, tiny little nucleus. So like I said, 10% of its function is clotting, but there is that other 90% that we don't really talk about, and that's really what that nucleus is tasked with doing. They are going to prevent fluid loss whenever blood vessels are damaged by producing some sort of clotting cascade or clotting mechanism so that we can plug the blood vessels so that blood can't leak out anymore. And these are produced from stem cells, from those mesenchymal stem cells. So let me write that out. Mesenchymal stem cell. And then those get differentiated into megakaryocytes. And then those finally get turned into platelets. So megakaryocytes. Okay, so how do we do this? How do we actually clot the blood vessel? So I think these are supposed to be arrows like this. That's what I'm imagining. Oh, sorry, like this. I don't know why it didn't capture that for whatever reason whenever I transfer them. Regardless. Step 1, right? You have to break the blood vessel. So for whatever reason, either you cut your hand or maybe it got brushed up against something with a pointy end to it, whatever. The blood vessel got damaged. And as a result, the blood vessel itself is going to spasm, freak out, separate itself, and then it's going to release chemical messengers that say, hey, help, I'm cut, I need help. And then platelets are going to come to the site, or thrombocytes. When the tissue is damaged, either the blood vessel is damaged or the surrounding tissue is damaged, or the surrounding outside of the blood vessel is damaged, those will also release chemical messengers called thromboplastin to recruit additional chemical messengers so that the clotting cascade can occur. So you have to remember that the blood vessel is a three-dimensional thing, a three-dimensional like tube. And you've got the inside of the tube that needs to be repaired and the outside of the tube that needs to be repaired. So some of the functions of the clotting cascade are going to happen within the blood vessel, and that's where the platelets are going to form. And then some of the clotting cascade is also going to function on the outside of the blood vessel, which some platelets are going to come to. But there's additional chemical messengers that have to be involved in this process in order for it to fully get plugged. So thromboplastin gets released, and thromboplastin is going to recruit calcium ions, which is super important. Calcium is really important in this cascade. And then some additional ions and proteins in order to help continue the clotting cascade. Then we're also going to have prothrombin that will be recruited. Prothrombin will also recruit calcium. and that will promote more prothrombin to come. And then prothrombin will eventually turn into thrombin. So anything with the word pro in it usually gets modified or cut into whatever the word is that follows after pro. So then we have thrombin. Thrombin is the active form of prothrombin. Thrombin can then recruit fibrinogen. Fibrinogen can become its active form of fibrin, and then we finally can produce a clot. So it's complicated, right? Like, this is like so many steps. Oh my gosh, what do I need to know? I would really focus on this slide for what items that you really need to know. Keep in mind, though, that as you take advanced anatomy and physiology courses, this clotting cascade is massive. It contains many, many different factors, many, many different types of ions, and lots of things that are activating other things that are nearby but not quite. So it's actually a really, really complicated process. This is probably the most simplified version of that process. Okay, once we create the clot, we have to have some mechanism of getting rid of it as well. So fibrin is going to talk to other fibrin, and they're going to create these strings of connective tissue to hold on to itself. So I think of it as like a platelet party. All the platelets get together, and fibrin is going to help hold together all of the platelets, and then they're all going to be at one party at one area and form a clot. And then at some point, we have to actually retract that clot. We have to get rid of it. And we call that fibrinolysis. Fibrinolysis, excuse me. So lysis is the breakdown of fibrin. And this is where the blood clot actually dissolves, right? At some point, we can repatch up the blood vessel. We don't need the clot there anymore. We can reuse some of the substances in the clot. And we don't want it to block blood flow anymore, so we need to get rid of it at some point. Thrombosis is where we have unwanted clotting. So thrombo from thrombocyte, cis formation of, and this is where we have clotting that wasn't supposed to be forming, right? This can be stationary or it can be moving. If it's moving, then we call it an embolus. And an embolus is really dangerous, especially if it gets into the lungs, the heart, or the brain. If it gets into the brain, then we can get a stroke, where the blood that was supposed to be supplying one part of the brain gets damaged and no longer functions anymore, and so that part of the brain doesn't have any oxygen going to it, so it dies off. Sorry, that's a plane in the background. Or we'll have, if it goes into the heart, then we might have a heart attack, where a part of the heart, again, stops receiving oxygen, can't contract anymore, it stops working. Or it can go into the lungs, which is also really scary. It can get lodged into a really, really tiny capillary, and then that part of the lung is no longer able to exchange oxygen with CO2 so that we can get oxygen to the tissues in the first place, and so part of the lung might die off. All very, very scary, all very, very dangerous. They need to be treated relatively immediately. But the point is, thrombosis is any type of clot, and embolus is a moving type of clot. Oh, I'm sorry, infarction. I didn't say this out loud, but this is a heart attack. Or, excuse me, heart attack is a type of infarction. You would say a cardiac infarction. Okay, here's a picture of the clotting mechanism, where vessel gets damaged, then you have some bleeding, then you have some recruitment of the thromboplastin. Thromboplastin turns into thrombin. Thromboplastin recruits prothrombin. Say this all 10 times fast. Prothrombin gets converted into thrombin. Thrombin can recruit fibrinogen. Fibrinogen turns into fibrin. Then you finally form a clot. All right, blood groups. How do we identify the difference between A positive, B negative, and so on? So there's all sorts of different types of blood types, and they're all predetermined, usually by genetics. And there are a variety of types, but not too many that there is not enough similarity, right? So we donate blood all the time, hopefully, and that blood has to be able to be recognized by the receiving recipient, right? We don't want the receiving recipient to have their immune system attack the donating blood. So there must be something to the red blood cell or the surrounding plasma that makes it either live in harmony with the recipient or not with the recipient. And so that's why we're going to talk about why blood typing is important. Whenever the recipient, let's say they don't receive the blood, or they receive the blood and they don't respond to it well, they actually try to attack it. What ends up happening is called agglutination. And agglutination is a clumping of red blood cells. And the reason why the red blood cells get clumped is so that they can form a target for the immune system to come and attack. And then the purpose of having the blood transfusion is useless because now we're attacking the donated blood. All right, what are the different types of blood types? We have type A, type B, type AB, and type O. And type A is going to be a red blood. Let me do this in red so that it's, there we go. We have a red blood cell, and type A blood is going to have A antigens. And in the plasma, it is going to produce anti-B antibodies. And let me just walk through this a couple of times and I'll recap here at the end. Type B blood is going to be a red blood cell with B antigens. And anti-A antibodies. Type AB blood is going to have both A and B antigens on it. And it's not going to produce any antibodies. Type O blood is not going to have any antigens on it, and it's going to produce both anti-A and anti-B antibodies. All right. Sounds like you said a lot of the same thing, Alexandra. What's going on? It all might sound alike, but they are very different. So let's focus here really quickly. Those are antigens. Antigens are going to be on the cell surface of the red blood cell. They are going to tell other red blood cells and other lymphatic cells, hey, I'm an A cell. I'm an A antigen. You like me, we're cool. Like, I was made, I'm supposed to be here, everything's fine, right? Or B, the same thing. Hey, I have B on me. Don't attack me. Like, we're cool. We're supposed to be B. It's okay. Or AB. Like, you like both of us. We're cool. We're supposed to be here. No problem. Or type O has no antigens. So if there's an antigen anywhere near it, it's going to go, oh my gosh, we should attack you. We should get rid of you. All right, what about the antibodies? So antibodies, there's a lot of anti here. Anti, right? So antibodies, let's just focus on those. Antibodies are going to target antigens, okay? Antibodies are going to attack antigens. So if we have an anti-B antibody, that means we are attacking B antigens, right? And this makes sense for type A blood, right? Because if we have A antigens on the cell surface, right, but no B, anytime we get exposed to B, we should probably attack it. So these are all going to attack B antigens. These are all going to attack A antigens. So why does type AB blood not have any anti-antibodies? Well, because it has both A and B antigens. So if we made any antibodies that were trying to attack A or B antigens, then we would die. So we don't have any anti-antibodies in the plasma of type AB blood. And then type O blood is the exact opposite. Since we don't have any antigens on here, we want to protect ourselves from both A and B. So we're going to attack both A and B antigens. Okay. All right. But Alexandra, I was told that I'm A positive or I'm AB negative. What does that mean? So that's all determined by the RH factor. RH factor. And there are many different types of RHs, but the primary one that we care about is antigen D, which determines if you're Rh negative or Rh positive, therefore A negative or A positive, et cetera, et cetera. And if you are, let's say you are Rh negative, if you are Rh negative, then you have never been exposed to the Rh factor because you don't have any of it. And so if you were ever exposed to the Rh antigens, then you are going to start making anti-Rh antibodies. If you were starting at Rh positive, right, now you've always made the Rh antigen, well, you're never going to want to attack yourself, so you're never going to make the anti-Rh antibodies. Okay, if that doesn't make sense, replay that a couple of times, okay? Now, why does that even matter, right? Not only with donating blood does it matter, but it really primarily matters in pregnant mothers. So let's say a mother is Rh negative, so she's never been exposed to the Rh antigen, but her baby is Rh positive, right? Genetics just said baby's going to make Rh positive. So A positive, B positive, AB positive, or O positive. Doesn't matter which one, but it's positive. Baby is making those Rh antigens. Well, those Rh antigens are really, really small, tiny dots, tiny, tiny dots. And those can pass through the placenta and go into mom's blood, okay? The blood isn't going in there, not the red blood cells, not the proteins, not just the Rh antigen is super, super small, and that can actually pass through the placenta. And then mom's blood is all of a sudden seeing Rh positive antigens or Rh antigens. And mom's blood is like, oh my gosh, well, I've never seen that before. I should probably attack it and kill it, right? But that's bad news bears because baby has that. And then mom's immune system would essentially kill off baby's blood. And then baby wouldn't survive. So this is why it's really, really important to have good prenatal care, because this has to be taken care of as soon as possible. And then you have to be visited again a couple months later so that you can have another dose so that you can take care of this so you don't attack baby. So when you go and you see the provider for the first prenatal visit, you'll actually be given, if this is your case, if you are Rh negative and baby's Rh positive, then you'll be given RhoGAM, which protects baby so you don't, I'm saying you as if you're the mother, so you don't stimulate your immune system to try to attack the A positive, B positive, AB positive, or O positive baby. If you were to attack baby and you didn't have this RhoGAM, it would be called erythroblastosis fetalis. Bad news bears. But if mom, let's say if mom is Rh positive and baby is Rh positive, then you're fine, right? Because mom is already exposed to the antigen, baby's making antigen, so all is well. It's totally fine. If mom was Rh positive and baby was Rh negative, you would also be fine because baby's immune system hasn't really quite developed yet. So baby isn't really going to respond to the Rh antigen if it comes over. And likewise, mom isn't even going to notice that Rh antigen isn't even existing in baby. It's only going to notice if something is there that isn't supposed to be there. So it won't attack it. It only really matters if mom is Rh negative and baby is Rh positive. That's the only time that it matters. Okay. Make sure you understand this concept. I know it can be really confusing, so go over it a couple of times until it starts to make sense. That's all we have for the blood system, though. Next, we'll go into the lymphatic system. See you all there.